The digital world brings an onslaught of data, opening tremendous opportunity to organizations globally. Yet this also brings challenges. What data can be trusted? How can data be made actionable? Organizations around the world trust Dun & Bradstreet for data, insights, and AI-driven platforms that guide intelligent actions and enable a competitive edge, addressing a range of use cases across public and private sectors, from startups to the largest enterprises, privately held and publicly traded. Dun & Bradstreet, helping you compete, grow, and thrive.
Hello again. I hope you enjoyed our first talk. Our second speaker on today's track is Ognjen Milicevic. Ognjen is an experienced bioinformatics scientist. He is currently working in the H-Tech group. Ognjen is also a teaching assistant at the School of Medicine at University of Belgrade. He holds degrees from School of Medicine as a general doctor and also engineering degree from biomedical engineering. His title of his talk is Budget-Friendly Sample Sizes for Genomic Research. After his talk, we will have a short coffee break and we will continue at 11:45. Thank you.
Hi, my name is Ognjen Milicevic from Belgrade, Serbia. I'm proud to be here on DSC 2022. Because of my mixed medical and engineering background, today I chose to tackle an interdisciplinary subject called budget-friendly sample sizes for genomics research. Let's start. We should open with a very practical question that I often get. Why do you need a biostatistician? Some of the common biostatistics tasks, from my experience, when interacting on a daily basis with medical personnel, is cleaning and transforming data to begin with from the initial database, then data description. All the assumptions and observation need to be verified by statistical testing. In order to present your data, you need to do some tabulation and visualization. And then maybe unexpectedly, bioinformatics is essentially applied statistics for genomics with a bit more engineering flavor sometimes. And then something that's especially important for my talk today, post hoc power calculations. When we say post hoc, we mean after the study is done, we try to verify our choices for sample size, for expected power, etc. There is also one very important thing we do on a daily basis. We complain that we weren't consulted earlier. We frequently have this feeling that if we were involved from the very start, that somehow we would make the study better, we would avoid some pitfalls, that our work would be easier if we were laying the groundwork for ourselves. But this is kind of unfair because for practical reasons, there is one main architect of many frequently conducted studies. And meet convenience, the architect of so many of the studies conducted today. This means that we are not choosing our samples in a random, non-biased way. We are actually selecting patients available to us, observation units which are convenient. And this results in some biases which have to be known, have to be explored. We have to bear this in mind when we are drawing conclusions. But essentially, this is not a major obstacle to our research. What it does kind of change the envisioned flow of research. We frequently do the post hoc sample size and power analysis, meaning that it's done after we have gathered some data and we have some numbers to work with. The reason for this is that we are gathering patients as we treat them. They do sign the release as they are usually treated in research facilities, so they agree to be included, but we don't really have an idea of how many patients we need, or sometimes we are not even sure what the conclusion would be. In that sense, we want to justify the choices we already made and the patients we already gathered. So how that works is we find a similar effect size in literature, effect size being the essential conclusion between your control and sample groups, usually. We use the posterior distribution as prior. So we are observing the patients and we are saying that this is our assumption. We are setting the desired power. This is a normal step for any kind of power analysis. It usually never goes below 80%. and then some people adjust as needed for dropout, any kind of losses that are expected. There is always some margin of error that can be assumed, and then all of that is used to obtain the sample size you already have, if that is in any way possible. In that way, you are validating the study that's already half complete. But it doesn't always have to be like that. This is kind of a be careful what you wish for situation, because sometimes people will include it from the start and ask you to make decisions which will impact the subsequent study. So, dear bioinformatician, can you tell us how many samples do we need to sequence to investigate the following problem? In that situation, there is no convenience there. Nothing is convenient about that. The whole problem is kind of like a black box to us. We're talking about sequencing, so this is something that's still fresh in a way. It's not routinely done. We almost never know the expected effect size, so what is the exact magnitude of the conclusion we are drawing. The literature is usually not helpful at this stage. There are multiple unknown genes that affect each problem. We don't know how many genes we expect to be relevant for this kind of a situation. The distributions of all the involved phenomena are quite complex. And then we get to the cost. Sequencing is frequently advertised as getting cheaper and cheaper, which is correct in the sense that it was unattainable to an average person some 20 years ago. But still, it's not cheap, even for research purposes. So I compiled some websites that transparently show their costs. It's usually not like that. You have to contact the provider and then ask for a quote. It's modified by many of the choices. But still, you can see that it's advertised for as little as $299, which if you browse the website, you will see that almost certainly what you require is much more expensive. Sometimes it's in thousands of dollars. It's very difficult to make some sort of range and distribution because there are so many choices to make there. But focusing on RNA sequencing at this point, you can expect to spend north of $500+ at any rate. And unless you're sequencing huge amounts of data, you won't get many discounts for bulk sequencing or anything similar. So let's talk about some aspects of RNA sequencing which are relevant for our data science-based analysis. I will briefly remind you that RNA is obtained by transcribing DNA, that it's most likely one of the most important steps of regulation, thus our interest in it, that there are more than 20,000 genes which can be transcribed, only a subset of them is transcribed in each cell, but still a very large number. And then we pose the question of individual distributions, so variations per gene. I outlined two papers here, both of them having a similar conclusion. First shows that less than 50% of genes are normally distributed, and then we have several other distributions to complement it. And then the other study finds that most genes can be modeled by the normal or log normal distribution, and the second most frequent would be the T distribution. So 99% of all genes can be modeled with the two of them. When we talk about the combination, the mixture of those genes, all 20,000 of them, we could maybe model them all together by joint distribution, which would be for each tissue some zero inflated distribution, because many of the genes in each tissue are not being expressed, hence the specific phenotypes of each tissue. But that is kind of not relevant for us because the expressions are not completely random and identically distributed. There are actually 20,000 plus separate outcomes. Then there is the next step. When we want to represent those RNAs from our cells with fragments, which is essentially what sequencing is, without going too much into details, we sequence many of the fragments, and then we try to map those fragments to the original genes. We quantify each gene, its transcription. We quantify it by counting all of those tiny fragments on the left. And the distribution that keeps arising in this kind of problem is called negative binomial distribution, or alternatively gamma Poisson distribution. This is for many applications for representation of DNA and RNA sequences. This distribution keeps popping up, and ultimately, if we need to choose one distribution to model this whole process, you will see that this distribution is chosen to do so. If we look at the whole pipeline now, we can see that there are three places where the variation can occur. There is the tissue sample and the whole process of sampling it. Then comes the step of chemical preparation, which is called library prep sometimes. That's the preparation for the third step, sequencing. The inter-individual variation in RNA, so the difference between individuals, that of course arises at the level of the biological sample. And this is where the effect between group comes. These two are the biological phenomena, and they are basically appearing at the initial step. In the chemical preparation step, we can observe many different effects, which depend on the chemistry being used, any kinds of contamination errors during preparation. Maybe the person preparing it. There are always variations for each technician that's actually working on it. And then there are so many other unobserved variables that we cannot even begin to imagine or measure. And it all comes in the step of chemical preparation, and we call them batch effects. This is why we prefer to have all our samples sequenced as identically as possible, possibly in a single batch, because that is supposed to incur least amount of unwanted variation. Finally, at the sequencing level, we have this representation variability, which is the number of fragments which will be observed from a truly random distribution. The output of this pipeline for a number of samples is a matrix of counts. This is kind of a transposed data science data frame in a sense that outcomes are placed into rows and all the samples are placed into columns. This is frequently complemented by a data frame of metadata, which is very relevant for tracking all the covariates. And these two pose an individual RNA expression experiment. This is all, as you can tell, layers upon layers of variability. So let's go back to those sample sizes. I feel it's good to talk in examples. I want to showcase a recent project which I've dubbed COVID-19 RNA characterization. The idea behind it was to explore the total RNA, both viral and human, in patients who were admitted to the hospital. So we've taken nasopharyngeal swabs and blood samples from them, and we opted for a paired design. So one sample of each of those blood and nasopharyngeal and swabs was taken on admittance, and then another was taken on discharge from the hospital. This is not a standard time difference, but at least 2 weeks or more after the admittance. The total was 18 individuals, so we had a total of 79 samples. And then we were posing the question, which biological pathways are affected? This analysis is called DEG, differentially expressed genes. And then what can we say about the viral load? And this is the branch called metagenomics, which we won't be discussing further here. So how do you estimate sample size for RNA? This task came to me, and when I started looking into it, the easiest and first way is you take some theoretical models with assumed distributions, which are usually negative binomial, and then you infer the parameters from previous datasets available offline. They don't have to be yours, obviously. This is all handled very neatly for you in many R packages. I named a few of these here. I haven't tried them all, but you can also, if you want a low-code or no-code solution, there's also a web tool that you can try where you have to decide on so many parameters there. So even if you have an out-of-the-box solution to do for this formula and this calculation, there are still choices to be made. Each of these choices gives a slightly different result, which means that you also get a distribution of sample sizes that's okay for you. And you know that they won't all work equally well. So if cost is not relevant for you, you can choose the most conservative one. So you make a number of methods, and then your ensemble prediction is the highest sample size that you got. But cost is always relevant. So the approach I'm proposing here now is you just perform one estimate and use it. This is kind of a statistically sound thing because you have no idea how to compare them all together. And then you remove any unwanted variability, which are actually batch effects. You reduce the variability with paired design, similar to what we have done. And then you use meaningful metadata to try to also explain some of the changes there. Filtering the genes is always useful. So in a way, you make sure that your power is sufficient by removing as much variability as you can. Let's go through all of these tasks one by one. So there is a number of methods, usually they're based on SVD, that try to remove batch effects without essentially tracing them to interpretable variables. You won't get an answer what happened, but you will reduce that variability. Also, these methods try to preserve the main effect by initially removing it, so they are advertised as being sound for your final conclusion. The two packages shown here, SVA and RAV-seq, they have kind of different approaches. One can also use some housekeeping genes which are kind of constant and easier to relate between samples. But a colleague of mine who I appreciate immensely said, once I see batch effects, I can correct them mathematically, but I'm never trusting that data set again. So this is kind of your choice if you want to go down this lane. I want to tell you that these batch effects are kind of the biggest problem of modern genomics, in the sense that we are sequencing so much data, we're making it available online. You can look in the SRA, which is kind of the main world repository for all kinds of sequences. There are 1,300,000 samples and more deposited there, but it's very difficult to include all of those samples together because of all of those batch effects. Sometimes they are explicitly done with a different technology, and that's okay. There are so many of them. You could still find some relevant for your use case, but those batch effects will simply swallow your effect sizes, and the conclusion will not be correct. For the pair design, which is kind of the main advice that I'm about to give, you basically take control samples from the same patients, either after resolution or before the event, if you can somehow predict it. This does increase power, but I must say that not all frameworks can take advantage of this pairing. It can be biologically difficult, and it does reduce number of degrees of freedom by half. We will discuss this in more detail later. For the meaningful metadata, I don't really have any specific advice. All of you from the data science background essentially know how this works. I can advise you that gender and age are always relevant, that you should always collect metrics of sample quality before and after sequencing to see if everything was performed correctly. You should also not mix disease subtypes, or at least mark them so you can subset them if needed. And then ultimately, if you don't plan to sequence all of the collected samples, use metadata in a data frame to select your samples. The filter genes is quite commonly known to everybody doing RNA sequencing, but you know that you're doing multiple testing. You're correcting for 20,000 plus genes. The multiple testing is severe. Any way you can get to a priori reduce the number of genes, it's very advisable. Just don't do it after the fact, because that's, cheating. We got a lot of different results. You can see an example on the left. This is from the nasal swabs for the paired case, which ultimately turned out to be the best. We wanted to compare the paired and unpaired cases, and for nasal, it didn't make too much difference. There were 40 genes passing the significance threshold for the paired design and 51 with the unpaired. So the number was decreased with the paired design. But then for blood, when pairing them, we got 76 differentially expressed genes and only 2 if we didn't pair them correctly. And then there are a few other parameter choices that I mentioned. We also tried to see the batch effects, to include them or not. And each of these choices kind of gives you a slightly different results. The p-values change, the rankings change. So how do you validate this? For the nasal, if you are just going for the number of differentially expressed genes, then you would say that pairing doesn't work well for nasal. What you can do is you can do annotation representation testing. There is an online no-code solution called PantherDB. For your information, annotation is any subset of genes which has like a common characteristic that's described. There are multiple available annotation sets. They tackle structure, function, pathway, whatever is interesting to you. They don't mix apples and oranges, but you can do each of them separately and review them. You only use significant genes and you don't use the p-values, you don't hack them. And then you can do two types of tests. You can do overrepresentation testing, which only takes a list of genes and it's essentially a chi-square to compare observed and expected frequencies. And then there is the enrichment tests. For that, you have to include a numerical variable, usually fold change, which is essentially your effect size per gene. And then you can do Mann-Whitney to test randomness of ranks. The most impressive result came from the molecular function annotations for the paired blood. As you can see on the left, I've bolded the interesting entries. You can see that the analysis has found that the immunoglobulin receptor binding was increased, since this is an infection. You can see that the olfactory receptor activity, that the reduction in smell is visible in blood. You can also see deodorant binding below that. Antigen binding is also related to the immune response, and then you can see way down, you can see two different categories related to oxygen binding, which we know is kind of severely damaged in the cases of bad COVID requiring hospitalization. When you find all of these categories, which you know are correct, you can say that your solution is valid, and then you can start exploring the other categories you obtain for novel discoveries. We consider the result validated by this. My takeaway would be that this study was rescued by pairing. This was a near miss because after analysis, we realized that we had no batches to correct. We didn't find any indications of that. We didn't have metadata available. And even though the signal in blood is smaller because it's a admixture of all kinds of signals throughout the body, you can see that it's very useful to track everything there. On the other hand, you can tell that the specific tissues, like the nasal tissue, gives out more robust results, so you don't necessarily have to pair it. So what happened by pairing from a data science standpoint? We reduce the individual variation by replacing the inter-individual with intra-individual variation. This is still significant in the sense that we respond to all the external stimuli and time of day and everything else with changes in our RNA. So we will still be seeing variation, but again, the magnitude is much smaller. What you also do is account for batch effects. This might not be completely apparent, but there is a good reason why that happens. See, pairing absorbs a proportion of batch effects. When you perform sequencing, you have many components which introduce some of their own bias. One of them is, for example, a flow cell. Flow cell has 8 lanes used for sequencing. It's ideal if you can always use an identical flow cell, but that's not always possible. It's much easier to focus on pairs instead of your whole groups of patients, because in that case you can agree that they were done, that at least pairs were done in a seemingly identical environment. When you do that, you also enable aggregation of data sets because those batch effects will be much smaller. We would be remiss if we didn't mention the downsides from a technical standpoint. You do lose half of your degrees of freedom, which can be relevant. You should know that not all frameworks can use this pairing, even if you have done it in such a manner. GLM is exceptionally easy to use in that way. There are many other tools which have their own empirical distributions, and you cannot do anything as a user to pair the samples. So what is the medical implication? Should we always pair? Let's recap the tissue conclusions. So nasal tissue had a much more robust signature and pairing didn't improve it as much. Blood does reflect many tissues. It naturally has a weaker signal because it's an admixture of signals from all around the body, but it does reflect all the local changes. You can see that even those olfactory changes are happening in your nose, they are reflected in your blood, which is fantastic. Some systemic effects live in your blood, some immune processes and things like that, and blood is always available for sampling. It's minimally invasive. From a utility standpoint, paired designs are easier to aggregate to meta-studies, which is especially important. They are robust to batch effects. Blood controls can be used as unpaired controls for other studies if they are healthy enough, so you can reuse them. And it does solve your problems of finding controls because your patients are also your controls. But we have to say that if controls are after resolution, which is frequently the case, the health is questionable. So for example, even in this case, long COVID is something that can mess up with your results and your interpretation of those people as healthy only 2 weeks after admitting them to the hospital. And then some or most chronic diseases cannot be caught early or they cannot ever be resolved. So the pairing is kind of impossible in that case. We will end with a few examples how to use this pairing in a seemingly unorthodox settings. Let's say we are interested in markers of atherosclerotic plaque progression or instability. So for example, you know that your patient suffers from them and you do patient checkup and sampling every X months, 6 months or something. You should know that sequencing is expensive, but sampling and storing is not as expensive. So if you do take a sample from your patient, each time he gets there and then you preserve it, you can also throw away the older samples. The important thing is that you are preserving the last 2 samples. If there is an interesting event for you between the 2 checkups, you can use the last 2 samples to see what has changed and what could be the markers of disease progression. I work a lot with neurodegenerative diseases, specifically ALS. If you're not familiar, it's the ice bucket challenge disease. Unfortunately, we cannot predict who gets the disease. It's familial only in 10% of cases, and the patient is available to you only once they are diseased. So you cannot get the normal control from that same patient. But if you sequence them sufficiently apart, you cannot find the root cause of ALS, because you're not catching the initial event essentially, but you can find what's going on while those neurons are deteriorating, which is the main phenomenon of this disease. You can find the markers of suffering and neuronal death, which is also an actionable point, because in a way, you can find a way to postpone or maybe even reverse this kind of damage. And this kind of generalizes well to all the chronic diseases. Finally, genomic analysis of cancer are almost always paired, but they are adapted for DNA sequencing. So you take the normal sample, which is blood, and then you compare it to the tumor sample, which is extracted during operations. If you do also take the normal sample for additional RNA sequencing, you can also benefit from understanding the expression landscape of this tumor. The problem with that is that you are already damaging somebody's organ by removing the tumor from within it. But this is always done with a margin of healthy tissue to prevent any kind of invasion. So you could theoretically sample this part and try to see if you can use that as normal. There is always the risk of tumor cells infiltrating this kind of area, and you would get an admixed signal. But still, this admixture can be overcome by all of those practices we were mentioning, so it does make sense. I would say I owe lots of thanks. So Institute for Bioscience and Medical Ecology here in Belgrade provided the samples and sequencing. My 2 affiliations, HTech Group, has provided computational resources and support, while School of Medicine has supported the research part. I really would like to thank the DSC organizer for inviting me here. And finally, I need to thank you all for listening. It was a huge pleasure relaying these experiences to you. Unfortunately, we don't have a Q&A session, but I would welcome any correspondence, feedback, or questions. Thank you all, and looking forward to seeing you again.